Now we are dealing with the structure of microspore or pollen grain. Now we are dealing with structure of microspore or pollen grain. So when we dealing about the pollen grain is the first cell of male gametophyte. It is first cell of male gametophyte. Pollen grain is termed as immature male gametophyte. It is immature male gametophyte. It measures, it is spherical in shape, spherical, it is spherical in shape and measure 20 to 30 micrometer in diameter. The pollen grain contains two layers. Pollen grain contains two layers, enzyme and exine. Enzyme, the innermost layer, inner layer, exine. The outer layer is thick, rigid and is composed of the outer layer. It is thick, rigid and composed of sporopollenin. Okay. So when we talk about the sporopollenin, it is a highly resistant, non-biodegradable and it is not affected by any other strong acid alkali. Okay. And when we talk about the enzyme, the inner layer, it is soft, thin and elastic in nature. Now we draw the diagram of pollen tube. And here the outer is exine. Okay. This is enzyme. Here the nucleus, tube nucleus, vacuole. This is generative cell. Okay. Now we start the labeling. This is exine. This is enzyme. This is tube nucleus, generative cell. This is germ pore. This is germ pore, vacuole. This is tube cell. And this is known as the structure of pollen grain. Now we are dealing with the development of male gametophyte. Okay. Now we are dealing the next topic, development of male gametophyte. So when we talk about the development of male gametophyte, we are dealing with the two stages. Pre-pollination development and the another one is post-pollination development. First, we are dealing with the pre-pollination development. In pre-pollination development, the development of male gametophyte. First, we are dealing with pre-pollination. Okay. In pre-pollination development of, first we are dealing with the term microgametogenesis, that is development of male gametophyte from pollen grains. Pollen grains develops in anther, so it is precocious or in situ germination. The cells of microspores divide mitotically to form two types of cell, that is large tube cell and small generative cell. Okay. So what happens here? This is the structure. Something is here. Further, what happens? The vacuoles are developed inside it. The large vacuoles are developed inside it. These are vacuoles and a green nucleus. This is a nucleus, which further show this here. This is asymmetric spindle, and here large vacuole, which further results this here a protective layer. This is vacuole, and this is known as vegetative cell, and this is known as generative cell. Now we are dealing with post-pollination development. The second topic is post-pollination. In post-pollination development, what occurs? It basically occurs on the stigma. Pollen tube absorb moist and sugar content from the stigma. Due to this, volume of internal content of cytoplasm increases. Volume of cytoplasm increases and exert pressure. It exerts pressures on both layers. Due to this, the enzyme comes out through germ pore in the form of pollen tube. In the form of pollen tube. In the form of pollen tube. Then, first of all, what happens? The vegetative nucleus enters into the pollen tube. Then generative nucleus enters and divides mitotically and forms two male gametes which are non-motile in nature.